morning. I'm Dr. Ingela Honey. Welcome to the last lectures for GTS 368. I'm a pediatrician by training, but I've been working for the past 22 years of my life with children and individuals with genetic conditions. I work on a daily basis requesting, interpreting, and explaining genetic tests. I do, however, not do the test myself, but will need the expertise of the laboratory staff to perform and partly analyze these tests. During the first section, we will be looking at chromosome abnormalities, which are traditionally divided into numerical and structural abnormalities. Numerical abnormalities will include all the trisomies and sex chromosome abnormalities. Structural abnormalities will include deletions, duplications, inversions, and translocations. Karyotyping were traditionally used, but it is now largely replaced by alternative DNA technologies. Karyotyping is still, however, used in certain circumstances. I will therefore go through the technique for completeness sake. When will you do an analysis of the chromosomes? It starts already prenatally when the karyotype will detect the presence or absence of chromosome abnormalities, such as in women that's over 35 years, known as advanced maternal age. Also when a couple previously had a baby with an abnormality, or if they themselves are carriers of structural chromosome abnormalities, such as balanced translocations. Where there's a family history of a specific condition, it might also prompt testing, abnormal antenatal sonar or abnormal serum screen, or an abnormality otherwise in the fetus will also prompt testing. If a mother is also very anxious of the possibility of an abnormality, it might be offered to her. Chromosomes can also be tested on the blastomere or a blastocyst in the case of pre-implantation diagnosis. I will, however, discuss this later on. Some conditions, if we can do testing directly after birth, it can help us to basically make, even before birth or directly after birth, it's possible to intervene. And we do chromosome analysis to confirm a clinical diagnosis. So a lot of medical disorders are associated with chromosome number or structural changes in children, infants, and even in adults. It can be used in cancer genetics because certain chromosome rearrangements and copy number variants is associated with certain cancers, and the prognosis can depend on the type of genetic defect. For instance, the Philadelphia chromosome in the case of AML. In the research setting, we will actually see that chromosome structure helped us to determine the function of genes. It also helped us to discover genes and map genes. So for this, I want you to actually have some prior knowledge. You need to know a little bit about the cell cycle with the different stages. Also need to know mitosis and the two stages of meiosis. Let's start with the human karyotype. What is a human karyotype? It is the chromosome complement visible during mitosis and best seen in metaphase. A cell with a nucleus is cultured and arrested during mitosis, examined under the microscope after being stained, and this will allow numerical and large structural changes to be observed. So what is the technique? First, let's start. Which cells can be used to be analyzed? So cells must contain a nucleus and therefore undergo division. Examples is lymphocytes, skin fibroblasts, bone marrow cells, and fetal cells in the amniotic fluid, the cord blood, or in chorioni villi, which is basically the premature placenta. The technique will then entitle cultivating the cells in tissue culture so that the cells can divide, and therefore the cell division is disrupted in metaphase. The cells are therefore thereafter put into hypertonic saline so that it can swell and basically, burst open so it can be spread on the slide. It's then fixed onto the slide and the chromosomes are released. You can thereafter stain it with DNA binding dyes. The most common we use is Giemsa, and thereafter visualized under the microscope. And this is an example of a normal male karyotype containing 46 chromosomes, consisting of a diploid number of chromosomes 1 to 22, and an X and a Y chromosome. Under the microscope, the following properties can be identified. The size and the structure of the chromosomes are determined by the centromere position. The centromere position classified the chromosomes typically into 3 types in the human. Metacentric, where the centromere is in the middle of the chromosome, so that the 2 arms are equal in length. The short arm is called the P and the long arm the Q arm. But this should be common information for you. And the submetacentric, the chromosome centromere is towards one end, so that there is a clear short arm and long arm. And in acrocentric chromosomes, the centromere is towards the tip of the chromosome, making the short arm actually not containing any expressed genes. Telocentric chromosomes is not present in humans, but that's where the centromere is on the tip of the chromosome. Chromosome 13, 14, 15, 21, and 22 is acrocentric. The banding pattern can also be divided, where the dark areas is AT rich and the light areas GC rich. The hemochromatoin pattern can be seen clearly on chromosome 1, 9, 13, 14, 15, 21, 22, and the Y chromosome. In this slide, you can see the banding pattern at different resolutions, depending on the condensate of the metaphase chromosome. In the case of high resolution banding, the different bands can be seen very clearly, enabling the detection of structural abnormalities. An ideogram is a schematic representation of the banding pattern of the different chromosomes. Chromosome structure is always described in a universal language according to the International System for Human Cytogenetic Nomenclature, ISCN. We use the chromosome number, thereafter the sex of the individual, the number of the chromosome that's dearranged, followed by extra and missing chromosomes. And here you can see a few examples of how we actually describe these abnormalities. I'm not going to go through it in detail, but you might need to identify some of these abnormalities. So what is the pros? What makes study a karyotype useful is that you look at the whole genome, and it gives an overall impression without any prior knowledge. Balance rearrangements can be detected, and it's possible to see polyploidies and mosaic cases. However, the resolution is very low, and about 10 megabases, 5 to 10 megabases is the lowest that you will actually be able to see on a karyotype if there's an abnormality. It needs culturing to get metaphases. It's very labor intensive and specialized, and very little automation. So a human being sits physically behind a microscope to do the test. It is time consuming, and it usually takes 3 to 14 days to get a result. You can imagine in the case of antenatal diagnosis, this can be a little bit long. The second method I want to describe is fluorescent in situ hybridization, also called FISH. And this basically also look at chromosome structure. So in the case of FISH, you combine a karyotype with hybridization techniques. The chromosomes or the chromatin is immobilized on a microscope slide. The DNA probes anneal to a specific DNA sequence. Probes are then labeled with fluorescent molecules, and the presence or absence of an abnormality can now be viewed under a fluorescent microscope. Here is the method a little bit more in detail. So you start with fixing your chromosomes on slides. You basically denature the DNA to single strands. You then will prepare your probe. It's a clone piece of the genome that has DNA sequence complementary to the area that you want to investigate. It's thereafter labeled with fluorosomes and denatured. These two are then on the slide hybridized, and you can now see the fluorescent signals against a background stain that binds to all the sequences that you have in your probe. Here is an example of a centromere probe. You can see the red where it's exactly on the centromere. The types of probes can be very locus specific, indicating the present, absence, location of a particular gene and sequence. Here is an example of the probe for the detection of a microdeletion on chromosome 22. One of your common microdeletions. Note that the one chromosome is a green control signal as well as a red signal, whilst the other chromosome, as you see in here, only have a green signal and the red signal is missing. The green control signal comes from the ARSA, the aryl sulfatase A gene that maps towards the locus 22q13.33. That's close to the telomere. And the red signal, which is basically the probe, matched to HIRA or histone cell cycle regulation or regulator, which maps very closely to your 22q11.2 area. You can use these probes for cancer, such as the HER2 amplified oncogene in breast cancer. Here is an examples of the MYC-N gene in neuroblastoma, and you can also pick up translocations in cancer, such as the BCR gene and the ABL1 oncogene in chronic myeloid leukemia. The Philadelphia chromosomes, where there's a translocation between chromosome 9 and 22. You can also manufacture repetitive DNA probes, which is actually aimed at centromere repeats or alpha satellites. This can be made very chromosome specific and can then detect copy numbers of a particular chromosome so that you can actually, for instance, diagnose a trisomy. And here is an example of a prenatal screen in interface cells, which basically detect trisomy of chromosome 21. Note on this side that you have 3 red signals. The centromere probes can pick up an additional chromosome 8 in a ring chromosome 8 in this preparation. You can also make whole chromosome probes. When that is done, it's called chromosome painting. This is very expensive and hardly ever used, and now replaced by another method. So the fragments is basically chosen that covers an entire chromosome or a chromosome arm. And here you can see an example of a single chromosome paint. In this example, a whole chromosome paint. There's a method called fiber fish, where chromosomes that's mechanically stretched is, hybridized to fish probes. In the case of quantitative fish, PEP nucleic acid oligonucleotides is used to target sequences, such as in this case of a subtelomeric probe, where it actually targets the telomere length or determine the telomere length. Again, each method has pros and cons. So the pros of fish is that it is actually can be used at higher resolution, and then both inter and metaphase chromosomes can be used. It also detects abnormal number of chromosomes, and it is quite a fast technique. You can also use different probes simultaneously. The cons, however, is that you do need prior information because you need to determine what is the area of interest, and the information you gain is really limited to that specific probe. And it's not a general screening tool. The third method I want to discuss is quantitative fluorescent PCR, abbreviated as QFPCR. It's now commonly used to detect aneuploidies. So it can be done pre and postnatal, and it specifically look at short tandem repeat markers on the chromosomes that's common involved in aneuploidies. That is chromosome 13, 18, 21, the X and the Y chromosome. So primers are then fluorescently labeled and analyzed by capillary electrophoresis. So hereby the technique a little bit more in detail. Any DNA can be used, cultured or uncultured, and you can use any type of cell, amniotic cells, chorionevae, blood, or buccal cells. The DNA is extracted. You then perform a multiplex PCR reaction with fluorescent labeled primers. These products is then separated through capillary electrophoresis according to the size. It's thereafter analyzed by software such as GeneScan or GeneMap. So the markers you use is highly polymorphic, short tandem repeats, and will be most informative if the markers exhibits different number of repeats on the 3 chromosomes. If not, you will have to do some quantification. If you have 4 or more markers, it's not difficult because there's enough information to make an accurate, result. In this example, marker data is interpreted from 3 common alleles that have 2, 3, and 4 tandem repeats. In the top trace, you can see the results is uninformative because the 3 markers is very similar and only 1 length variant is detected. In the middle trace, the result is fully informative. You can see that there's 3 different markers and you have 3, traces. In the bottom trace, you have to do some calculation. It suggests a trisomy as the one variant is twice the size as the other. So that basically indicates that you might have a trisomy because the 2 length variants will indicate that there's 3 chromosomes. In this practical example, the output shows traces of 3 of the markers. You can see it's blue and green and black. And that represents assays for microsatellite markers on chromosome 13, 18, and 21. The control X and Y. This result strongly suspect a trisomy 21. As you can see that there's 3 signals for this specific locus and 2 signals each for this specific locus, of which one is double the size of the other, representing 3 chromosome 21s. The top marker, or this one, is those 2 are informative, but this specific one is uninformative as you only see 1 trace. In this graph where they actually analyze the markers, you can see that again, this is a trisomy, as this one indicates there's 3, and this also indicates 2. And you can clearly see that those lengths are rather equal, and these are actually one a little bit higher than the other, 28 to 12. and again in this one too. It is a little bit more difficult in the case of sex chromosome abnormalities, as it's more varied, and it's sometimes very difficult to distinguish a monosomy, a tetrasomy, or even a pentasomy X. Counting the sex chromosomes is, however, possible by using primer sets that are simultaneously amplified on both sex chromosomes and on an autosome. Look at your figure 11.3 in the textbook for further information. It's a little bit complex. I won't ask you questions on that. Again, what is the pros and cons of qFPCR? The pros is that you can use any nucleated cell if it's cultured or uncultured. The results are rapid, and you get it usually within 24 to 48 hours, and there is many short tandem repeats available to use for multiplex PCR. It can even be used in the case of non-invasive fetal aneuploidy screening, and it's highly accurate for a specific chromosome. It's less labor intensive. The cons, however, is that it only screens for the common aneuploidies, and no information is gained on any other structural or numerical abnormality. And very often it's used with other diagnostic procedures, such as following with a karyotype if normal. The last method I want to explain is your chromosome array. So why would you do a chromosome array? It's to detect deletions and duplications less than 5 to 10 megabases of DNA, which you won't detect any abnormality using chromosomal banding techniques. It's a microarray based DNA assay, and it can scan copy number, that is deletions and duplications at the submicroscopic level. There's two types of microarrays. The one is your array-based comparative genomic hybridization, and the second one is your single nucleotide polymorphism microarray. In the case of array CGH, your patient or your test DNA is labeled with one color, green, and the reference or the normal DNA is labeled with another color, red in most cases. These two samples are then mixed and hybridized to DNA probes on a slide or a chip. The ratio of red to green fluorescence is then measured along the length of each chromosome. The amplified DNA in the patient will then show increased copies and hybridize stronger to normal chromosomes if it's increased, and you will get a more green signal. If there's a deleted DNA in the patient, there will be less copies and it will hybridize less to the normal DNA with a more red signal. So let's explain it. So you have your two DNA populations, which you've labeled with green and red. Fluorophores, green. The samples are then mixed and hybridized to DNA probes immobilized to a single chip. The single chip is called a microarray. These probes are long oligonucleotides and usually about 55 to 66 base pairs. On the array, you will now be able to distinguish between normal and abnormal. So the ratio of the red here and the green here can now be analyzed and further, usually with computer software. So the arrays can basically cover the entire chromosome, called the whole genome arrays, or it can be targeted, such as in the case of microdeletion syndromes, just subtelomeric. It's very common use for genomic abnormalities in cancer, submicroscopic aberrations, or prenatal genetic diagnosis. It can therefore also detect chromosomal imbalance in cancer for these large-scale changes, and in this case, the tumor samples is usually compared with the DNA from the same individual. It's also very commonly used to look for suspected gene imbalances in children with developmental delay and congenital abnormalities. I use it very often in my practice, and it's now the first tier test in these patients. Here is an example for an array CGH picking up a trisomy 21. You can see there's some loss in the X but gain in the Y, so you can see it's a male individual, and here you can see there's gain in chromosome 21. You will, however, not use an array to determine Down syndrome, as you will use a QF-PCR or a karyotype rather. Here is an example of a male infant with global developmental delay, has got dysmorphic features, broad thumbs, and heart abnormalities. In this child, the child's DNA was labeled with CY3 and the control with CY5. It was therefore hybridized to an array with 60,000 different oligonucleotides. The small green probe, you can see it here, the small green probes represent individual oligonucleotide probes on the vertical scale, marking the position on chromosome 60, from position 1 to 90 million. The red and the green lines mark the limits for the normal. You can see the red and the green lines. The red bar on the left here marks for a deletion, and the green bar on the right, duplications. So here on the ideogram, you can see they've indicated the deletion, and the deletion is indicated by this specific nomenclature, and there is the different base pairs. That's only one copy, and the duplication on chromosome 16. You can see the two different duplications. You can see how they've actually written it out. And there's 29 known genes in this duplication and 170 known genes in that duplication. So lastly, SNP microarray hybridization makes use of panel of single oligonucleotides that's shorter in length than those in an array CGH, as they only hybridize to individual alleles. So for a single SNP locus, the oligonucleotide will be present to only hybridize for that specific locus. Here, the SNP arrays is not compared to a patient's tests, to a control sample, but to a database of SNP array results from a lot of control individuals. It will also pick up losses of gains across the genome, but it will also detect absence of heterozygosity. In this case, because of the absence of heterozygosity, you can detect a deletion. So there will only be one of the two alleles. In duplications, it will vary, however. Use, for example, if you have a SNP locus for two alleles, A and B, a heterozygote will just have an A and a B. But in case of a partial trisomy, it might be either AA and B or A and BB. The use of SNP arrays to identify gains and losses are a little bit more insecure, and array CGH is usually used rather for this purpose. You do, however, get commercial microarrays where both components of an array and a SNP array is done simultaneously, and the Afrometric genome-wide human SNP array has about 946,000 long oligonucleotide probes to detect copy number variation and short oligonucleotides for typing of alleles at more than 906,600 SNP loci. This is an actual case. A little girl was referred to me at about 20 months of age. When I saw her, I was told that she had a QFPCR at birth, indicating a trisomy 18. It was unusual because babies with trisomy 18 don't usually live up until 20 months of age. We decided it might be a mosaic, so we did chromosome analysis, which came back normal female. I therefore questioned the QFPCR, but when I repeated it, it again showed a trisomy 18. I therefore knew there must be extra chromosome 18 material and asked for an array. And this was my result. I'm not going to go into detail. It was done in Leuven a few years ago before it was available in South Africa. Here you can see the gain on chromosome 18. I detected loss of material on chromosome 21. So the results indicated that there was a tiny duplication on the X chromosome that was maternally inherited, that they regarded as non-pathogenic. There was also a tiny paternally inherited deletion on chromosome 17, also regarded as non-pathogenic. But what was regarded pathogenic was a large duplication of chromosome 18 and a large deletion on chromosome 21. When we re-looked, we looked at the father and he had an 1821 translocation. This just tells us that you can't, and the interpretation of these copy number variants is very significant. So you have to decide, is it abnormal or pathogenic? So when do we decide? It's when it's been described in syndromes, when it comes from, when it's not inherited from the parents, or if it's identical to an affected parent. When it's very large and there's a lot of genes involved, it can also be regarded as pathogenic. If it's homozygous and a deletion, or if there's a greater than one copy gain, it's amplified. Very often you get unclassified variants and incidental findings, and we luckily can refer to databases. It's always important to do parental samples to see if the variant is present in the parents, because if it's in the parent and the parent is normal, it might be not the cause of the abnormality. So if it's de novo, this interpretation might be very difficult because there might be just not enough data so that the variant will be unclassified. Also very important is that because it scans the whole genome, you can get incidental or secondary findings, where findings unrelated to the reason of testing can have important health consequences, such as adult onset cancer syndromes. That is usually a dilemma and need to deal with care. If it's likely benign, it's maybe previously reported to not be any significance, and it's inherited from a healthy parent. It can be identical to a healthy parent. It might be that it's described in healthy individuals, but also the duplication might not have dosage sensitive genes, and there might be no regulatory elements. So all this information is obtained from databases such as Decipher. But there's still, in some cases, conventional karyotyping necessary, because the arrays can't detect balanced chromosome rearrangements where there's no loss or gain of DNA. As you can see here, this is a balanced translocation, so there's no loss or gain of material, and the array won't pick it up. It also won't pick up inversions. But the pros: whole genome analysis, very good resolution. You can do it blindly. The gains and the losses can be detected simultaneously. And in the SNP array, you can detect uniparental disomy. However, the cons: it's very expensive, can be very difficult to interpret result. It can't detect balance abnormalities, as I mentioned, and it cannot distinguish between insertion, a duplication, or a marker chromosome, or unbalanced translocations. You can't determine the chromosome localization, so the duplication can be on a different chromosome, and it very often can't detect mosaic state. The resolution in chromosome visualization will determine the technique. So for G banding, you can pick up 1 to 10 megabases. For fish, 500 kilobases to 1 megabase. Fiber fish, it's a little bit more sensitive, for 5 to 500 kilobases. Array CGH, 25 to 500 kilobases, and a SNP array signal nucleotides. So it's very important to decide which test is the most appropriate for your patient, and that is my challenge. So I hope this gives you a good overview of all the different chromosomal techniques to determine abnormalities.